In this video, I want to show you the testis. This is the structure of the testis. Why? Because it is made up of a series of tubules that have been cut in different ways in cross-section. These tubules are the seminiferous tubules, and because they are highly coiled. When sectioned, they may appear in longitudinal cross or oblique sections. Each testis is externally covered by a connective tissue capsule called the tunica albuginea. This capsule is made of collagen fibers as well as fibroblasts. It's called the tunica albuginea because the collagen fibers are collectively white, which makes this part appear white when unstained. If we look beneath the tunica albuginea, this area is loose connective tissue that sends its extensions into the testicular tissue and forms a tissue called the interstitium. The areas I am showing you here are the interstitial tissue. Inside the interstitium, many sperm-producing tubules or seminiferous tubules can be seen, which appear in different cross-sectional shapes. The seminiferous tubules are lined by a specialized epithelium called the germinal epithelium because it plays a role in forming germ cells. Sperm are the male germ cells. The lining of the seminiferous tubules is made of two types of cells: supporting cells or Sertoli cells, whose nuclei you can see here, and the spermatogenic cell line. The spermatogenic cell line includes cells called spermatogonia, which are located at the base of all seminiferous tubules. This dark, round nucleus belongs to one of the spermatogonia cells. Spermatogonia are of two types, type A spermatogonia and type B. Type A spermatogonia undergo successive mitotic divisions and produce a large number of type B spermatogonia. Type B spermatogonia, in turn, enter the first meiotic division. During the first meiotic division, the cells enter prophase, and the chromosomes begin to condense. From this point on, this cell is referred to as a primary spermatocyte. These nuclei, which are actually the largest nuclei present in the seminiferous tubules, are identified as primary spermatocytes. These are primary spermatocytes. Primary spermatocytes have nuclei with a dotted or thread-like appearance. The reason for this is that the chromosomes begin to condense in these cells, and as a result, we can observe cross-sections of the condensed chromosomes. The number of primary spermatocytes in the seminiferous tubules is high. The reason is that first meiosis is very long in duration in males, and therefore their number increases in the seminiferous tubules. Primary spermatocytes gradually enter the second meiotic division. A cell that enters the second meiotic division is called a secondary spermatocyte. The second meiotic division occurs rapidly. Therefore, the number of secondary spermatocytes present in the seminiferous tubules is very low, and we may not even be able to see them in tissue sections. Secondary spermatocytes are morphologically similar to primary spermatocytes. However, they have smaller nuclei and are located closer to the lumen. Secondary spermatocytes gradually transform into cells known as spermatids. These small cells with pale, round nuclei located near the lumen are identified as spermatids. Spermatids have completed the stages of meiotic division and therefore have haploid nuclei containing half the number of chromosomes. After that, they undergo differentiation. Their nuclei become more condensed and elongated, like the ones you can observe here. These nuclei transform into the condensed nuclei of sperm. In addition, a flagellum appears in these cells. And the acrosome also forms at this stage. In this way, mature sperm are produced. They are located toward the lumen and are ultimately released into the lumen. These are the sperm that have been released into the lumen of the seminiferous tubules. In addition to the cells of the spermatogonial lineage, other cells are also present. For example, this cell, or more precisely, this nucleus is the nucleus of a Sertoli cell. These nuclei also belong to Sertoli cells. The nuclei of Sertoli cells are large and triangular in shape, and they may have a prominent nucleolus in the center, as you can see in this area. These nuclei are usually lighter in appearance and are located near the base of the seminiferous tubules. Their cytoplasm is not clearly visible, and its boundaries cannot be easily distinguished because it contains numerous folds that accommodate the cells of the spermatogenic lineage. For this reason, we cannot easily observe the cytoplasm. However, the cytoplasm extends from the base all the way to the apex, or the lumen. Behind the basal lamina of the seminiferous tubules, elongated nuclei can be seen, which are known as myeloid cells. These elongated nuclei that you see here are identified as myeloid cells. Myeloid cells are located behind the basal lamina of the seminiferous tubules, and their contraction can play a role in propelling sperm forward. Between the seminiferous tubules, we observe the interstitium. This is the interstitial tissue, which is composed of connective tissue. In this connective tissue, in addition to collagen fibers, you can also observe fibrocytes. In this area, there are also several specialized cells known as Leydig cells. Here, you can currently see 3 Leydig cells next to a capillary. The structure visible here is a capillary, and inside it, there is also an RBC. Leydig cells have the characteristics of steroid-secreting cells. Here again, a cluster of Leydig cells is visible. Leydig cells have centrally located nuclei and acidophilic cytoplasm. Their acidophilic cytoplasm is due to the abundance of SER and numerous mitochondria. The SER and mitochondria in these cells are responsible for the production of a steroid hormone called testosterone, which is considered the primary male sex hormone. This hormone is released into the blood vessels. In this image, you can see numerous Sertoli cells. These Sertoli cells play an important role in the formation of the blood-testis barrier. I would like to show you these Sertoli cells, which are seen here with pale nuclei and prominent nucleoli using a three-dimensional model. In this 3D model, you can observe the structure of Sertoli cells, the spermatogenic cell lineage, and also Leydig cells. The cells shown in gray represent Sertoli cells. As you can see, the nuclei of Sertoli cells in this area appear oval or triangular in shape, and they are pale. Their cytoplasm is so extensively folded that it can accommodate the cells of the spermatogenic lineage within it. Above the spermatogonia, which are located on the basal lamina, we can see the extensions or processes of Sertoli cells. These processes extend toward each other and connect via tight junctions, thereby creating two separate compartments. One compartment near the lumen and another compartment farther from the lumen. Within the compartment that is away from the lumen, you can see the spermatogonia, which have been present since birth. However, after puberty, these cells divide and give rise to pre-spermatocyte cells. These cells were not present at the time when the immune system was established in the body. Therefore, the body may produce antibodies against them. As a result, the body must protect these cells. The protection of these cells is achieved by the processes of Sertoli cells, which are firmly connected to each other through tight junctions. As a result, primary spermatocytes are protected from exposure to antibodies that may reach them through the blood vessels. These primary spermatocytes divide too, form secondary spermatocytes, and then spermatids. Ultimately, spermatids undergo differentiation and produce sperm. At first, the sperm remain attached to the Sertoli cells, but later they are released and float freely within the lumen. Behind the basal lamina, Leydig cells can be seen. Leydig cells are polygonal cells with a centrally located nucleus and acidophilic cytoplasm.